All right, so welcome to our second video where we're discussing the nervous system. So we finished up, or with the first video, after we had discussed neurons, structure of neurons, kind of the overall makeup of the nervous system, and kind of how it's broken up into different divisions. And we looked at what action potentials are and how action potentials are kind of transmitted throughout a neuron through that saltatory conduction. In this second video, we're going to look at how transmission happens from a neuron to another target cell, whether it's another neuron, muscle, or tissue. And then we'll also look at the peripheral nervous system. So we'll talk about the specific components and divisions of the peripheral nervous system. And then after that, we'll get into our third video where we'll talk about the central nervous system. And so to start this video off, we're just going to look at how information is transferred from a neuron to its target. And like I mentioned, that target can be another neuron, it could be a muscle cell, it could be any kind of tissue cell, organ cell, or gland. And basically what happens is at the end of the axon terminal, there is a synapse area. So this synapse area is basically a gap or special junction where a neurotransmitter will move across that junction and bind to the target cell. Okay. So let's say you have like an axon, and then that axon will have an axon bulb, and then let's say you have a muscle cell. So this area is called synaptic cleft. Okay? And the transmission from the neuron to the muscle cell basically involves these little transmitters, so little molecules that will move from the axon terminal across the synaptic cleft to the target cell. And so that impulse will be sent across that gap to the receiving cell, whatever that cell is. And like I mentioned, this involves the release and diffusion of a neurotransmitter. It will diffuse out of the neuron terminal across that synaptic cleft and bind with the target cell. Okay? And so when an action potential arrives at the axon terminus, what that action potential does is it causes calcium to diffuse into the axon bulb. So the bulb that's at the terminus of the axon. The calcium will then cause the neurotransmitter to release from vesicles. Talked about exocytosis, where vesicles combine with the plasma membrane of the cell and push material to the intercellular environment. So that's basically what's happening here. So the neurotransmitter will be in a vesicle, and then it will be pushed across the plasma membrane into the synaptic cleft. So basically the gap between the neuron and the target cell. The neurotransmitters will then diffuse across that synaptic cleft and bind to receptors that are on the target membrane of the target cell. And then those receptors will cause, or those target kind of proteins that the receptors bind to will open gated channels that allow sodium to diffuse through the plasma membrane and initiate a graded potential that will start an action potential in that target cell. Okay? So you have your action potential propagating down in this figure. That causes calcium to be released when it gets to the axon bulb. That calcium then says, okay, the vesicles with the neurotransmitters will release those neurotransmitters into the synaptic cleft. So you can see the bulb here, the target cell here, and then this gap here is the synaptic cleft. Okay. And so if we zoom in to this area, is what we see here. And so you have your synaptic cleft up here. You have your neurotransmitter that was released from the axon bulb, diffuses across the synaptic cleft, and then binds to the protein that has a binding site for that neurotransmitter. When the neurotransmitter binds to that protein, it opens that protein and allows for that sodium to come through, causing a graded potential in that target cell. And then depending on the cell, a specific response will happen. If it's another neuron, then that action potential will carry on. If it's a muscle contraction may be initiated, et cetera, okay? And so again, the response depends on the target cell. The response of the target can involve several different types of neurotransmitters. There's over 50 types of neurotransmitters that can be released depending on the target. That means there are many, many different types of receptors to receive those neurotransmitters, as well as many different types of gated ion channel. Now you can have two types of neurotransmitters. You can have excitatory neurotransmitters, which will depolarize the postsynaptic cell, causing it to go over the threshold, right, and have an action potential, right? Or it could be an inhibitory neurotransmitter, basically causing hyperpolarization, so it doesn't go over that threshold, and it basically inhibits a response from that target cell, basically stops it from doing so. You can have neurotransmitters that are excitatory or inhibitory. Okay. This figure here comes from your table, and it actually tells you some of the more known neurotransmitters, so like acetylcholine, serotonin, endorphins, you're probably familiar with, norepinephrine. Tells you where these sites are released. So for example, neuromuscular junctions, which is where muscle contractions happen. Serotonin is in the brain and the spinal cord. Endorphins, many areas in the brain and the spinal cord. And then it tells you what are some actions that happen because of that. So like acetylcholine is important for movement of skeletal muscles. Serotonin is important for like mood, sleep cycle, etc. So a lot of these neurotransmitters are very, very important for allowing our bodies to function appropriately. Okay. So we have a little animation here that's kind of going through that process of transmission from the axon terminal across the synapse to the target cell. And so we'll just take a look at that.
Chemical communication between two neurons occurs across a short gap between the cells called a synapse. An action potential generated in the presynaptic neuron travels down that neuron's axon to the synaptic terminal. This signal is then transmitted across the synapse via chemical messengers and is picked up by the dendrites of the postsynaptic neuron. These dendrites then convey the signal to the cell body and axon of the postsynaptic neuron, where the action potential is regenerated. When the action potential reaches the synaptic terminal, it triggers an influx of calcium ions. This influx of calcium triggers the fusion of synaptic vesicles in the synaptic terminal with the terminal membrane. These vesicles contain a chemical neurotransmitter that is then released into the gap between the synaptic terminal of the presynaptic neuron and the membrane of the postsynaptic neuron. The neurotransmitter molecules diffuse across the gap of the synapse, where they bind to the postsynaptic ion channels, causing the channels to open. If the synapse is excitatory, the flow of ions through these open channels depolarizes the postsynaptic membrane, and this depolarization is propagated along the membrane of the dendrite and eventually into the cell body of the postsynaptic neuron. If the signal is strong enough, and depending on what other signals are being received by the postsynaptic neuron at the same time, another action potential will be generated when the signal reaches the axon of the postsynaptic neuron. Once a neurotransmitter is used, it must be cleared from the synapse. One way to remove neurotransmitters from the synapse is via reuptake. In this case, the neurotransmitters are transported back into the presynaptic terminal and reused. Another way to clear the neurotransmitter is via enzymatic degradation. In this case, the enzymes degrade any neurotransmitters left in the synaptic gap.
All right. One interesting thing about the kind of response of neurons and the target cells is that the postsynaptic cell, so the cell that is receiving the information, the response from that cell actually depends on how many neurons that it's forming a synapse with, and whether or not the neurons forming those synapses are excitatory or inhibitory, right? So that means, it's either going to cause a response or prevent a response. Now, there are two kind of setups that neurons can have. You could have convergence, where one neuron receives input from many others, or divergence, where one neuron sends action potentials to many other neurons. And so here's just kind of an example of that, where we have convergence. So you have, multiple neurons bringing information to one single neuron. So all of those signals are converging into one place. And then we have, after that, an example of divergence, where you have one neuron sending one signal, and that signal is diverging into four different postsynaptic neurons after that one, of four different directions, basically. So again, that's convergence and divergence. So that's how action potentials are transmitted to their targets. And like I mentioned, the response really is determined by the neurotransmitter, how many neurons is interacting with that postsynaptic cell, and then what exactly that postsynaptic cell is, whether it's muscle or another neuron or some other cell that's inside the body. What we're going to look at now is the peripheral nervous system, and we're going to talk about what its job is. And its job specifically is to relay information between the tissues and the central nervous system, and then from the central nervous system to the tissue. And so whenever we say nerve, we're really referring to axons